should say that, yeah, despite the affiliation, I'm not a mathematician, so I should apologize in advance for the very hand-wavy things I'll say. So in this talk, I hope to discuss how the conditions for local adaptation in a subdivided population, focusing especially on the effects of polygenic architectures of local adaptation. So, when we think of subdivided or patchy populations, we might have some sort of mental image of, a number of patches, each sort of connected to each other in some complex way. And these may be sort of subject to different local environments. And one would like to understand how local adaptation depends on, various factors such as, how these local environments are distributed across patches, how these patches are connected, whether what is the genetic architecture of local adaptation, whether local fitness is a sort of polygenic trait or something that's determined by a handful of loci. And of course, we might be interested in a number of other factors such as mating system and biotic competition and so on. And of course, in its full generality, this is a complex problem, but we can always start with sort of a caricaturized model. And this is sort of a special case of the model that Nick actually talked about yesterday, so I'll maybe go through it quickly. So this is, so I'm going to consider an infinite island model where each island is connected to every other island via migration. Every island has a fixed number, n, of haploid individuals. And so our population size is constant and independent of local fitness. So this is the so-called soft selection limit. In each generation, a fraction m of, this is quite bad, no? Okay, in each generation, would it be, maybe should I just remove it or? Otherwise people on Zoom. Okay. So in each generation, a fraction m of individuals are replaced by migrants drawn uniformly from Thank you. So in each generation, a fraction of m of individuals on each island are sort of replaced by migrants who are drawn uniformly from the entire metapopulation. So they can be drawn from any island with equal probability. So, this is essentially a model of subdivision, but there's no space, no concept of near or far. We have two habitats and a fraction with a fraction row of islands belonging to one habitat and a fraction, the remaining fraction to the other habitat. And I will consider sort of habitat dependent fitness, where local fitness is influenced by L unlinked loci. So by unlinked, I just mean that they segregate independently of each other. So the model that I'm considering is one where there is divergent directional selection across habitats. So if we have, a biallelic loci, so a loci where there are two alternative alleles. So in that case, we have alternative alleles favored at each of those L loci across the two habitats, independent of the state of the other loci. And for simplicity, I'm also going to consider equal effect loci, where, and in that case, the relative fitness of an individual who carries X locally deleterious alleles is just exponential time of minus S times X. for the most part, I'll neglect mutation because we're interested in understanding how sort of mixing between populations influences local adaptation. Except, so I should say that I'll to sort of illustrate some of the ideas, I'll focus first on this special case where one of the one of the habitats is vanishingly rare. In that case, the common habitat essentially evolves independently of the rare habitat, and in the absence of mutation, it should essentially fix the alleles that are favored in that habitat. So this sort of rho tending to zero limit essentially becomes a mainland island model where the mainland is perfectly adapted and there's maladaptive gene flow from the mainland to the island. And because we're interested in how local adaptation actually influences sort of genetic exchange between populations, not just at loci that are implicated in local adaptation, but also at arbitrary locations along the genome. I'm also going to consider divergence at unlinked neutral markers, or which often there are various measures, for example, FST, that one could use to quantify this. So just to sort of put it all together, we have a model where the parameters are rho, which is the fraction of islands in the rare habitat, L, which is the number of divergently selected loci. Then we have S, M, and 1 by N, which one can think of in the continuous time limit as sort of rates of change due to selection, migration, and drift, respectively. I've sort of considered unlinked, that is independently segregating loci. So the rate of recombination between any two loci, whether selected or neutral, is just half. And this is, as we know, this basically means that associations in the absence of other forces, in the absence of selection and other forces, so associations or pairwise LD would decay at a rate which is half per generation in this case. So, the sort of analysis of even so caricaturized a model in some sense is challenging basically because of two features. One being that we have multiple loci under divergent selection. So if a locus comes in, a deleterious allele that sort of immigrates into a population will not immigrate on its own. It will typically be associated with other deleterious alleles, and thus it experiences sort of indirect selection. So in other words, we have to consider multi-locus evolution explicitly. We have to consider the effects of LD. The other sort of source of complexity is that we are thinking of a subdivided populations where individual populations may be small, so we need to account for drift. And a lot of analyses, often, I mean not all, but very often we typically neglect one or the other complication. So, for example, what Nick presented last evening was a linkage equilibrium analysis where we neglect LD. Conversely, you can sort of neglect drift and write down deterministic equations for allele frequencies or for genotype frequencies. And so but of course, to understand this, we would need to sort of put both together in some way. So I'll start by basically introducing, or rather summarizing the sort of linkage equilibrium and deterministic approximations, and then indicate how one might, combine these in a very heuristic way. And then I'll sort of illustrate these briefly for the mainland island model and then also the infinite island model. So, yeah. So just it's sort of useful to think about these limits in terms of, the relative rates of different processes. So in a situation where recombination, the recombination rate, which is half, is sort of higher than all other rates, or that is, recombination is faster than all other processes, we have a situation where loci essentially evolve independently of each other. So this is the linkage equilibrium case. In particular, what we require is that LS, which is the net selective disadvantage of migrants coming in who carry deleterious alleles at all their L loci. So we require this LS to be significantly smaller than R, which is of order 1, or basically half. In this case, we can think of loci as evolving independently, and one can work out the distribution of so allele frequencies at each locus, and this is of course well known for a very long time. It was so this was worked out by Wright, and we know that in the diffusion limit, what matters is just these scaled parameters nm, nmu, and ns. Mu is I should say it's the rate of mutation, which we need for the mainland island case. And of course, one can extend this to the whole metapopulation case, which was already something that was worked out by Wright. And yesterday, I guess you saw the hard selection extension as well, where one can work out the joint distribution of population sizes and allele frequencies. And of course, the key point is that for small nm, we have essentially U-shaped distributions where loci are close to fixing one or the other allele. One can also sort of think of the other limit where we have a sort of drift that is slower than all other processes. In particular, we require drift to be weaker than selection at individual loci, such that even individual deleterious alleles essentially follow deterministic evolutionary dynamics. And in that case, we can write down deterministic coupled equations for the genotype frequencies, where these frequencies are, these probabilities or frequencies are essentially correspond to genotype frequencies of genotypes having 1, 2, up till L alleles. These are quadratic equations and quadratic coupled equations, which are, of course, not so easy to work with. But in the limit where migration is rare and deleterious alleles are present at low frequencies in the population, one can sort of linearize these equations and work out the genotype frequencies and work out the average allele frequency, which is basically the average number of deleterious alleles per locus. So one can sort of work this out. It's essentially just algebra. But it's sort of useful to think about this in a specific limit where the total divergence is due to a very large number of loci of weak effect. So what we are doing essentially is keeping LS, which is the net maximum selective disadvantage, constant, while taking the L tending to infinity, S tending to 0 limit. And in this limit, one can sort of show that this essentially to the 0th order in S is, reduces to the simple expression, and of course, and one can, work out the first order correction and second order correction and so on, which is just a Taylor expansion. So in this, for these polygenic architectures, what one finds is that the net effect of LD on deleterious allele frequencies is captured essentially by having something like an effective migration rate, which is reduced relative to the raw migration rate by this factor e to the minus 2ls. And this is just, the reproductive value of migrants in this limit, in this whatever, L tending to infinity, S tending to this particular limit where we imagine the total amount of divergence to be due to a larger and larger number of loci. So this is, of course, also for neutral alleles, this is also the effective migration rate. So essentially, what we have is that in the thinking about a deterministic population, gene flow at whether it's at a deleterious allele or at a divergently selected locus or at a neutral locus is essentially attenuated by a factor equal to the reproductive value, which is essentially the long-term genetic contribution of the migrant to the population. So this is again sort of known, but we would ideally like to think about a situation where we drift is significant enough to perturb the level of local adaptation, at least at individual loci, and where the net selective disadvantage, which is given by LS, is not necessarily that small, because if we are thinking, for example, about speciating populations, we might be looking at rather high levels of reproductive isolation, or rather low reproductive values of migrants coming in. So we have a situation where drift is comparable to migration and selection, and LS is comparable to sort of R. But even in this situation, there is a natural sort of separation of time scales that emerges because we have polygenic architectures. So we sort of see that drift, can't be much stronger than S. Drift has to be of the same order as S to sustain local adaptation at individual loci. But that means that it has to be significantly weaker than selection at the level of genotypes. So this sort of suggests that we could combine a deterministic description of LD with a stochastic description of allele frequencies, and see how it goes. So that's my hand-wavy justification for doing this. So the sort of really simple-minded way of doing it is to ask whether if we just introduce effective migration rates into Wright's formula, into this single locus diffusion approximation for the distribution of allele frequencies, how does it do? But of course, one has to then ask, what is this relevant effective migration rate? So we know that, I sort of argued that this is proportional in, this S, the highly polygenic limit. This is just attenuated with respect to the raw migration rate by this e to the minus 2sx. E to the minus 2sl. But of course, this is true only in the, if you think of the previous slide, it's this sort of assumed that, that the deleterious alleles are rare enough for us to ignore the effects of their effects on, let's say, the mean fitness, or where essentially the fact that we can linearize our genotypic equations for our genotypic frequencies boils down to that, among other things. And but we would like to actually be able to do this not just when deleterious alleles are rare, but also when they're common enough for local adaptation to collapse. And sort of one justification for one way of doing this, at least in the limit where NM is weak enough for allele frequency distributions to be U-shaped, basically for us to have nearly fixed selective differences between populations, is to see that what is determining the so our so when we have a situation where the expected allele frequency is half, this would essentially correspond to having a 50% of loci close to fixation for the locally beneficial allele within the population versus 50% close to fixation for the deleterious allele. So the effective number of loci in some sense is not L, but L time, the number of loci that actually differ between the two populations. So what I'm going to do really is just use my deterministic expressions for effective migration rate. So not the approximate ones, but basically the full ones, but with an effective L, which is not the total number of divergently selected loci, but the number of loci which actually sort of might be expected to have these fixed differences between the two populations. So one can then sort of work out the expected allele frequency. This has to depend on, so you end up with a sort of transcendental equation really, which has to be numerically solved because the expected allele frequency depends on the effective migration rates, which are themselves a function of the expected allele frequency so in this mainland island case. And doing so the last step involves a sort of numerical solution, but overall, what we're doing is we are sort of abstracting the effects of all this multi-locus LD via a single effective migration rate, which depends only on the expected allele frequency in the population, expected level of divergence between the mainland and the island. So, so I'd maybe in the next 5 minutes, I'll just briefly describe how well this does. So in this plot, you see the expected frequency of the locally favored allele on the island versus the average number of migrants per generation, that's NM. The symbols are simulations, the lines are the sort of the theoretical expressions where we have to numerically solve for in the last step for the expected allele frequency. The different colors correspond to different L, with fixed S, so basically different values of LS. So one thing, of course, is we see this remarkably good agreement between full individual-based simulations and, this simple heuristic of incorporating effective migration rates into the diffusion approximation. We also see that as we go towards larger and larger values of S, or larger values of L, this sort of loss of local adaptation becomes more abrupt, which is something we expect when there's sort of this LD-driven elimination of deleterious alleles. One can also do this one can also ask whether this is a regime in which drift is making any difference at all. So here I show the same thing, but now the different colors correspond to different values of N. The symbols are simulations, the solid lines are the sort of approximation, which accounts for both LD and drift. The dashed lines are the linkage equilibrium approximations for a given value of N. And the dotted line is the deterministic prediction. So we see that we are in a regime where drift strongly reduces the critical migration rate at which local adaptation is lost. So this is, as we go from the blue to the brown curves. LD is also, the other thing is that LD is much more significant in larger populations. So this is this sort of sharp threshold that you see for where NS and NM and Nmu are sort of larger. And that all of this can be captured by the simple heuristic of having effective migration rates in our diffusion approximation. One can also predict other things, such as the whole allele frequency distributions. And finally, this also extends quite well to the metapopulation case, where now instead of we sort of need to have consider whether have different effective migration rates for migration within the same habitat versus migration between different habitats. For migration within the same habitat, there is no attenuation of the effective migration rate, but for migration between different habitats, we sort of again assume that this is effectively reduced by the average divergence between the two habitats, and then one has to numerically solve for the average levels of divergence. And this is, again, just to say that it sort of does very well. These are different parameters. On the right, you have, on the left, you have a situation where 10% of islands belongs to one habitat, and on the right, where 30% of islands belong to the other habitat. And one sees these protective effects of LDR stronger in the case when one of the habitats is very rare. So I will not go into this except, and I'll just briefly mention that one can also, incorporate these effective migration rates into a structured coalescent framework to obtain the average levels of, to obtain FST at neutral loci. And one sees this rather striking behavior where FST of the rare within the rare habitat for Deems within the rare habitat is much higher for FST for Deems within the common habitat, as long as there is adaptive divergence between the two habitats. And this can also essentially be sort of rationalized by noting that, since most individuals migrating into the rare habitat originate from the common habitat, they have an attenuated sort of effective migration rate, so that the net effective immigration into the rare habitat is low. This results in lower heterozygosity within the rare habitat and higher FST. So I'll sort of end here and just mention that I think, beyond the simple model, what this seems to suggest is that there's this nice way of bridging what we know of, trait level properties such as reproductive values, which often can be expressed in a simple, in terms of just the trait divergence and the trait variance. And so this is work by Nick and Allison, with this sort of very locus level or genic level description of differentiation by just this simple heuristic of including effective migration rates into the diffusion approximation. It would be nice to sort of understand when it's justified. I mean, I indicated this by I justified this in terms of, thinking about a fixed state approximation where different alleles are fixed, where different loci are essentially fixed for different alleles. But actually, another justification is that when you have, a Gaussian distribution of trait values, then the reproductive value essentially depends on the trait mean and the trait variance. And one can, so in this limit where S tends to 0 and L tends to infinity, it's just the average difference between trait means that sort of matters. So I'll stop here.
Okay, thank you very much, Himani. And is there any questions online? Or?
Thank you very much for the very nice presentation. If I understand well, you in your justification there is this U-shape behavior for the fixation probability, and in some of your very first figure, it was not clear that it was satisfied for every set of parameters. My question is, do you have some kind of limit or critical parameter at which you lose this shape so that somehow your theory sort of reach some limit, and what does happen in this case, and are you able to identify this limit?
Yeah. So yeah. So that. Sorry. So that, yeah, as I said, that's once actually it's very hard to find parameter regimes where this doesn't work. So I wasn't able to find such parameter regimes, at least for this unlinked case. And this also works, for example, when the distribution, so this is the distribution of allele frequencies at very large NM here, which is, it's definitely not U-shaped. And so I think the other justification is that this is a paper by Nick and Allison a few years ago, where they sort of work out reproductive values of under the infinitesimal limit, where they show that this is so if you have if you imagine an island population with trait mean Z bar and variance variance V, then this, and if you ask what is the reproductive value of a migrant with trait value Z, so then this depends on and so beta is the strength of directional selection on the island, and there are terms which involve the segregation variance, which is related to the trait variance in this infinitesimal case. And the limit that I'm just taking, so they're assuming a Gaussian distribution of the trait value. So that's very far, that need not be, so that's not this fixed state approximation at all. And the limit that I end up taking here right now is for SL constant. And in that limit, basically these terms don't matter. It's just this. So that's also, so I think this approximation holds more generally. And in fact, one could also work with their full expression, which would be, I think, although they don't derive reproductive values for stabilizing selection, but in the case of stabilizing selection, we would want to worry about the variance terms in the reproductive value. And, yeah.
Okay, thank you. Any other questions?
I was wondering if you could use the same approach to a different setting where instead of local adaptation, you would have some DIMs with the harsh environment where the deleterious alleles may have stronger effects, and DIMs with say a more permissive environments where the deleterious alleles would have weaker effects. And so you would have an increased effective migration rate from in one way. And
I haven't tried to do that, but I mean, I think as long as, so if you're imagining unlinked loci, and in that case, so we would still, so for example, this kind of justification should work, I think, for in that case as well. Yeah.
We will thank again Himani and